Diagnosing COVID-19 requires taking a sample from the patient, usually using a nasopharyngeal or oropharyngeal swab, or more recently, a saliva sample, and testing for the presence of viral RNA using real-time reverse transcriptase polymerase chain reaction, or RT PCR. So how does RT PCR work? The first step is to extract and isolate all the RNA from the patient's sample. But RNA is more difficult to work with in the lab than DNA, and DNA can be amplified and quantified using a relatively simple, accurate method called the polymerase chain reaction, or PCR. But PCR doesn't work with RNA. So we convert the RNA to DNA using an enzyme called reverse transcriptase. The resulting DNA is known as complementary DNA, or cDNA, because its sequence is complementary to that of the original RNA strand. Now, it's important to know that at this stage, we've actually created a collection of cDNAs that represent all the RNA that was present in the original sample. This could include RNA from bacteria or other viruses, or from the patient's own cells. So we need a way to determine whether SARS-CoV-2 cDNA is present in the sample. For that, we use PCR, which allows us to amplify and detect a specific DNA molecule, in this case, the viral cDNA. PCR involves 3 basic steps which are repeated upon 40 times. In the first step, all the double-stranded DNA molecules are denaturated, meaning the 2 strands are separated. In the second step, a pair of primers that are specific to the SARS-CoV-2 cDNA anneal or attach to each DNA strand. The primers are short nucleotide sequences that are complementary to a unique sequence in the viral cDNA. The specificity of the primers ensures that they only bind to the viral cDNA and not to any of the other cDNAs present in the sample. In the third step, called elongation, an enzyme known as a polymerase adds nucleotides to the ends of the primers, using the original DNA strand as a template, and now we've gone from 1 double-stranded DNA molecule to 2. These 3 steps, denaturation, annealing, and elongation, are then repeated with the amount of DNA doubling in every cycle. So if you started with only 1 cDNA molecule, after just 35 cycles, you would have 2 to the power of 35, or over 34 billion identical DNA molecules. In real-time PCR, a probe is added during the PCR process, which gives off fluorescence whenever a new DNA molecule is formed. The increase in viral cDNA can be followed in real time by tracking the increase in fluorescence signal. When the level of fluorescence exceeds a certain threshold, we can be confident that the signal is significantly above the background level. The CT, or cycle threshold, value is the number of cycles required for the fluorescence signal to exceed that specific threshold value. The lower the CT value, the more RNA was present in the original sample, indicating a higher viral load. For SARS-CoV-2, a CT value of less than 40 is considered a positive test. If the result is positive, it is also important that the CT value be reported, since this indicates the patient's viral load or infectiousness. Several studies have shown that viral load, as measured by CT value, can help predict disease progress. For example, this study of hospitalized patients showed that those who developed more severe disease had lower CT values on their initial RT PCR test on admission than those who developed more mild disease. They also showed that the probability of disease progression correlated with the CT value on admission. Therefore, as a clinician, you would not only want to know if your patient was positive on RT PCR, but also by how much. What was their initial cycle threshold value? Because as we have seen, a patient with a cycle threshold value of 10 has 1 million times as many viral particles in their throat as compared to a patient with a cycle threshold value of 30. Obtaining quick, accurate test results is important to prevent transmission of the virus. So how long does RT PCR take? A typical real-time PCR run takes approximately 2 to 4 hours, including the time it takes to extract and reverse transcribe the RNA. The specific equipment available and the level of automation of the process is key to determining how quickly a specific lab can produce results. Because time is of the essence during a pandemic, many companies have developed newer RT PCR kits that can produce results in as little as 15 minutes, although many of these still need to be validated. Obtaining quick results is great, but it's just as important that these results can accurately identify people with the disease, so they can be isolated to reduce transmission. So, what's the accuracy of RT PCR testing? To answer that question, we need to consider both the analytical specificity and sensitivity, that is, the ability of the RT PCR assay itself to detect the viral RNA when it's present in the sample, as well as the clinical sensitivity and specificity, which tells us how well the test detects individuals who have or don't have the disease. It's the clinical specificity and sensitivity that ultimately determines the rate of false positives, individuals who test positive but aren't actually infected, and false negatives, individuals who test negative but are actually infected with the virus. The good news is that RT-PCR has a high analytical and clinical specificity for SARS-CoV-2, meaning that it produces few false positive results. So when a patient tests positive for the virus, we can be fairly certain that they are actually infected. When they do occur, false positives are generally the result of technical errors or reagent contamination and are generally avoidable with good laboratory technique and the use of proper testing controls. The analytical sensitivity of SARS-CoV-2 RT-PCR assays is still up for debate, but it's generally thought to be high. However, the clinical sensitivity of RT-PCR for SARS-CoV-2 is only around 70 to 80% at best. One study estimated it to be as low as 38%. This means a single negative test result does not always mean the patient doesn't have the disease. False negatives are a big problem during a pandemic, since these patients actually have the virus, but they think they're in the clear, so they may engage in behaviors that cause them to transmit the virus to others. But what could be causing these false negatives? Although most RT-PCR kits use primers that target regions of the viral RNA that don't appear to change much, variations or mutations in the viral RNA sequence could lead to false negative results by preventing proper primer binding. And not all SARS-CoV-2 RT-PCR test kits are created equal, with some being more sensitive than others. But ultimately, any RT-PCR assay is only as good as the sample put into it. So the sample collection process is probably the biggest contributor to false negative results. This could include improper sampling technique, transport, or storage, but the type of specimen used and the timing of sample collection most likely have the greatest influence on the overall sensitivity of SARS-CoV-2 RT-PCR tests. We will examine this in more detail in the next